This is an overview of the OncoPanel service offered by Eurofins Discovery. OncoPanel is a flexible platform for the evaluation of a wide range of anti-cancer therapeutics. We can accommodate the testing of small molecules, biologics, both conventional and designed subdomain proteins, as well as things like aptamers and potentially other therapeutic types. We currently have more than 300 genomically characterized human tumor cell lines in our collection, and we run through the testing of those 300 plus cell lines every 3 months. Studies of different sizes can be ordered, but we'll talk about that a little later. In the OncoPanel collection of cell lines, we have a broad cancer type and subtype representation, which covers a range of different tumors, different subtypes of tumors, and a range of different genetic and genomic compositions. This is afforded by the fact that we have genomic characterization of the vast majority of the cell lines that we have to offer. Genomic characterization has been taken from both the Cosmic Sanger and the Broad CCLE databases, wherein we have actually taken the downloads from both of those publicly available databases, normalized and integrated the databases to form what is effectively a proprietary internal database. This allows us to interrogate drug performance against the OncoPanel of panels typically larger than 200 cell lines, and to be able to correlate drug performance with genomic composition of different tumor cell lines. All of the cell lines that we use are obtained from reputable sources, typically the ATCC, although we do have some cell lines from the DSMZ in Europe, as well as the Riken collection in Japan. We have a very large cell bank, which affords consistency in performance. This also gives confidence that if we do a study with a client at the current time, and then we perform a study a little later, we will typically be using cells from the same bank or same working stock of cells that were made for the initial study. This allows comparison between different studies and makes that a lot more affordable in terms of confidence, as well as being able to correlate the data between the two studies. The service has been in operation since 2009, and for every plate of every cell line that we run, we typically run a DMSO control as a vehicle control, as well as a sporosporin reference compound control, which we test over 10 different concentrations. We monitor and track the performance of every cell line in every study that we do, and consequently, over the 10 years of operation of OncoPanel, at the current time of recording this overview, we have data wherein we know how each cell line should be performing in its normal state in these assays. We have validated assays for a variety of different exposures, which I will talk about in a few minutes. But we track the performance of each cell line by measuring its number of doublings in a defined period of time, as well as the IC50 and the EC50 for that reference compound sporosporin. For a cell line to pass QC, we need to show that each cell line has a doubling and a sensitivity to sporosporin that is within acceptable error margins of historical data. If a cell line performs aberrantly, and any one of those 3 different factors, doubling time, EC50 for sporosporin, or IC50 for sporosporin, are outside of those acceptable error margins, we will fail that cell line, and we will repeat the testing of that cell line at cost to us. We also do not culture cells for extended passages of time due to the inherent ability of cancer cells to genetically drift. This is another level of quality control, and again, another facet of the platform that gives high confidence when comparing between different runs done at different times. The OncoPanel platform provides a number of different assay formats from which to choose. We have formats that are monitored by high content analysis, as well as traditional viability assay formats. We have assays that are validated for exposure times of 3 days and 5 days, with up to 305 human tumor cell lines currently being available. We also have a validated assay for a 10-day exposure, with 275 cell lines currently available. This assay was developed in order to meet the needs of the epigenetic drug discovery community and is also applicable to other compounds and other test agents with different mechanisms of action, but which require extended exposure times in order to be able to see their full effect. This includes things like the Aurora B kinase inhibitors, as well as PARP inhibitors. Because we use high content analysis in the above-mentioned assays, we can offer a singleplex assay, which is simply a proliferation assay. In this assay, we denuclear stain the cells at the end of treatment. For every study, we are also running a T0 plate. So this allows us not only to calculate EC50 and IC50 values, but also GI50 values, which we report for all proliferation assays, along with the activity area or the integrated area above the curve. We are able to multiplex other readouts with the proliferation assay readout, and that is our standard multiplexed offering. In this offering, we not only offer the standard readouts for the proliferation assay, but we also offer the ability to interrogate test agent performance as to their ability to induce apoptosis, which we measure by evaluating levels of cleaved caspase-3, as well as their effect on the cell cycle, which we evaluate by measuring levels of phosphohistone H3. In both of these cases, we compare the responses to the vehicle control, and for the induction of apoptosis, we report the concentration at which a 5-fold increase is achieved over the vehicle control. For the cell cycle read, we report the concentration of the test agent that is required to give a 50% change or a 2-fold increase in the phosphohistone H3 signal. This correlates with a G1S arrest for the decrease or a G2M arrest for the increase, and that is also relative to the vehicle control. For studies with more than 200 cell lines, we offer a genomic analysis of the data, and that is part of the service. We can offer genomic analyses for panels of fewer than 200 cell lines, although we do not necessarily encourage this, because the sample population size of 200 cell lines or more has been found to provide a particularly robust, statistically relevant analysis or significant analysis. When we do a genomic analysis, this identifies a number of predictive genomic biomarkers for both sensitivity and resistance towards the test agent, which can have tremendous use in terms of directing activities beyond IND. I'll talk about that in a few minutes. As with many of the assays available, we can customize these assays upon request. So please contact the Eurofins discovery team for more details around that. The OncoPanel platform can also offer 3D spheroid models, and we have a number of models set up for different cell lines using a 3-day 3D assay. And this is currently with 120 different models that are available. These assays are evaluated using a viability format. This uses cell titer glow 3D, and often can be run in parallel with a parallel 2D assay. The 3D assays can be run independently, but there is also the ability to compare with 2D in cases where the client would like to do so. As with the 2D assays, there is ability to customize the 3D assays in various ways. And again, we are happy to discuss that with the client depending upon their needs. The OncoPanel platform can also be used to evaluate drug-drug combinations. We can do this with all of the above-mentioned formats. And we conduct a traditional 9x9 combination matrix, wherein drug A is evaluated at 9 separate concentrations against 9 concentrations of drug B. We compare those combinations with the single agents alone, and we provide excess over Bliss indices for all of the combinations. We present these data as a heat map, so one can see areas of different combinations that correlate with either synergy, or additivity, or antagonism. We also provide the individual curves for each of the combinations. We do have a series of standard of care data sets that are available that were generated in the standard OncoPanel assays, be they singleplex or multiplex. And those are also available for purchase. But with our experience using standard of care agents, we are also able to work with the client to provide rational and logical combinations with their particular test agent in order to provide value for their program. We also offer a panel of normal cell types comprising 7 different human primary cell types and 3 immortalized normal human cell lines. These span a range of different tissues and can be useful in counter-screening for agents that show activity in the above-mentioned OncoPanel assays. We do perform other services. We have the capability to do extensive cell banking. This may be useful for activities beyond IND and especially regulatory studies. As I've mentioned previously, we can do custom studies not only with the formats listed above, but with other formats. For example, we have conducted custom studies in single cell lines wherein we are using high-content imaging to look at one or more cellular markers in response to drug treatment. We can also customize exposure times and other aspects of the assays. In terms of the numbers of cell lines in each panel for a particular study, we can conduct studies ranging from small to large panels. This might be the full set of cell lines that are available for any one particular assay format, or this may be a single cell line for a very specific application. We have, as I mentioned previously, a broad tumor type and tumor subtype cross-representation. Panels of cell lines can be selected based on tumor type, or panels of cell lines can be selected based upon genomic composition. For example, we can put together panels with the client or design panels for the client that represent aberrations in a particular gene of interest, whether that be overexpression or mutation status. We can identify predictive genomic biomarkers, as I mentioned earlier, by taking primary data, evaluating the drug response, and then rank ordering the cell line response to that particular agent for the parameter being examined. As I mentioned, for the proliferation assays, we can evaluate EC50, IC50, GI50 values. And for apoptosis, we measure the 5-fold induction over vehicle. And for cell cycle, a 2-fold change, whether it's an increase or a 50% decrease in the phosphohistone H3 signal. We can conduct genomic analyses for all of the proliferation readouts and the apoptosis readout. Unfortunately, we cannot calculate or we cannot conduct genomic analyses on the cell cycle readout for the simple reason that the signal can be biologically relevant where it is increasing in one cell type, but also biologically relevant where it decreases in another cell type. As a result, where you have an increase and a decrease that are both biologically significant, it is very difficult, in fact almost impossible, to call relevance for biological markers and genomic markers that are associated with a change that can go one way or the other. However, with the other parameters, we can group cell lines into resistant groups, and those would be shown towards the right of the graphs, the waterfall plots that are shown. And we can group cell lines into sensitive groups, and those will be shown at the left of the above-mentioned graphs. We can then conduct statistical analyses of those groups to find out what genomic properties correlate with those groups. We conduct orthogonal statistical assays to rule out the bias that is inherent in any one statistical analysis. So the data and the results that we obtain are driven purely by the biology and the particular cell line's response to the test agent, and not by a mathematical bias of any one analysis. We identify mutations that correlate with sensitive immune resistance. We also identify differential expression patterns that correlate with sensitivity and resistance. And collectively, these genomic analyses provide a lot of value for programs, and especially late-stage programs as one is moving towards clinical trials. In those cases, this can help facilitate patient stratification by providing an indication as to which mutation types or which expression profiles within a patient's tumor might render them potentially sensitive and thus would obtain benefit from treatment with the particular therapeutic agent in question. These genomic analyses can also be useful from the tissue type cross-section in identifying broader utilities of an agent beyond that for which it may have originally been designed. This can provide other tumor types based on tissue that may not have been initially targeted. And it can also identify other genomic compositions of cell lines or cell types, tumor types, that render them sensitive, that again might not necessarily reflect the primary target to which the drug was designed, but nevertheless render the cell type and thus the tumor sensitive. These analyses can also be useful in identifying intrinsic resistance mechanisms, which again allow one to address this early by doing some of the combination studies that we discussed earlier. In terms of drug discovery, typically going from early to late stage drug discovery, the number of compounds is very large and then ultimately filters down to the one compound or the one test agent that is going to be selected to go forward into clinical trials. Against the backdrop of that, assays typically increase in complexity. From assays with maybe a small number of cell lines early on in the drug discovery process, wherein a large number of compounds can be filtered for those that show most promising activity, through to models and assays that are more complex in design, but more interrogate the properties of a smaller number of compounds and ultimately the candidate in much more detail. So consequently, as much is known as possible about that candidate before taking it into clinical trials. This is shown in the progression on the current slide from high-throughput screening through the hit-to-lead process, through lead optimization to pre-IND, and then to IND, with the appropriate milestones of declaring leads and selecting candidates. So from the perspective of OncoPanel, small panels are often beneficial early on in the drug discovery process, and we are quite happy to design small panels that meet the client's needs, or to work with the client to design those small panels and test larger numbers of compounds or test agents. In those cases, this is fairly easy for us because all of our process is automated. For lead optimization, we can provide both regular screening against smaller panels or periodic screening against larger panels where broader utility is beginning to be interrogated. And for late-stage programs, as I've mentioned previously, we can provide much larger panels, including full panel selections and genomic analyses, to thoroughly characterize agents before IND application and clinical trial entry. Consequently, we can help with the finding, refining, and the defining of agents as one goes through the drug discovery continuum. This is a function of OncoPanel that we can use in both the proliferation, the mechanistic side of evaluation of test agents, as well as some of the custom assays that I've mentioned earlier that might include things like looking at a PD marker or other cellular markers of relevance to the program. OncoPanel offers tremendous innovation, and as I think you've seen, flexibility, as well as a very high-quality service. We have one of the largest cell line collections available. We have currently, as I've mentioned, a little over 300 human tumor cell lines available, which is more than many of our competitors, but not necessarily the largest. However, it is large enough for us to be able to conduct meaningful and valuable analyses for all manners of drug discovery. We offer higher precision of our data because we offer 10-point dose response curves from testing every agent in triplicate. We can, of course, customize this if a particular client would like to look at fewer concentrations, and especially in some of those earlier stage applications that are more of the screening nature. We do, as I mentioned, offer several different assay formats, including 3D models, providing a more comprehensive evaluation of test agents as one refines and develops those into more drug-like molecules. We do have a multiplexed option, as I've mentioned earlier, which helps not only address whether an agent inhibits the proliferation, growth, or viability of a particular tumor cell type, but it helps address how the agent is manifesting that change. We also offer the genomic analysis, which is a key differentiator and provides the values that I outlined on the previous slide. As I've mentioned periodically throughout the presentation, we have tremendous project flexibility in what we can do. While we do have platform offerings, we are very keen to work with the client to provide solutions that meet their exact needs. That can sometimes include looking at different subsets of cell lines, and we are quite able to customize cell lines and add additional cell lines to panels that we may not currently have in the collection and run them as custom cell lines, again, to meet the client's needs. We have flexibility in the assay formats, as I've mentioned. We can change the exposure time, sometimes the assay conditions, and other parameters, again, upon discussion with the client, pending the practicality of being able to do so. We can also use our platform technologies for other assays, not necessarily of an oncology application, but in particular, in relevance to oncology, as I've mentioned, we can use the high-content imaging system for evaluation of changes in PD markers and other cellular markers in a phenotypic context that may be variable upon treatment with a particular test agent. And again, if this provides meaningful data for the program, then this is something that we are quite happy to work with the client to achieve. If you have any questions about the service, please contact Alistair King at Eurofins Discovery with the following contact information. Thank you very much for your time and attention.